हेलो फ्रेंड्स, वेलकम टू ए टू जेड सब्जेक्ट। ये पेपर हमको CBSE बोर्ड online.com से मिला है और CBSE बोर्ड online.com पे सारे CBSE के पेपर और उनके सॉल्यूशंस प्रेजेंट हैं। आप वहाँ जाके चेक कर सकते हैं। तो आज हमारे पास है बायोलॉजी का पेपर 57/5/1 सेट 1 12th क्लास बायोलॉजी का CBSE का पेपर हम सॉल्व कर रहे हैं। सेक्शन ए, फर्स्ट क्वेश्चन, identify the parts PQRS in the given figure below and select the option in the correct order. So B is the right answer. First is the thalamus, second is seed, third is endocarp, and fourth is mesocarp. So B is the right answer. The fetal ejection reflex in human triggers the release of which particular oxytocin and maternal pituitary. B is the right answer. A DNA molecule is 160 base pairs long. It has 30% guanine. How many adenine bases are present in this DNA molecule? So the answer is 64. In Pisum sativum, the flower color may be violet or white. What proportion of the offspring in a cross of VV cross VV would be expected to be violet? 100%. In a human female, menstrual cycle of 28 days is represented by the diagram given below. Here both A and B are correct. The primary follicle of ovary matures into graphene follicle in phase 2. The hormone secreted in large amounts in phase 3 is also responsible for maintaining pregnancy in human females. So both A and B are correct. Arrange the following in the correct sequence of their evolution and select the correct option. C is the right answer. First, invertebrates. Second, jawless fish. Third, seaweed. Monohybrid purpurus is a yeast used commercially in the production of which one of the following? Statins. Given below is the restriction site of a restriction endonuclease PST1 and the cleavage sites on a DNA molecule. The right answer will be D. Choose a correct option that shows an interaction in which one species benefits while the other is neither harmed nor benefited. C. Anemone and clownfish. B. Transgenic animals have their genetic material manipulated. Select the correct option in reference to the transgenic animals. Foreign DNA is present in all of its cells. C. Select the odd option in the context of convergent evolution. Thorns of bougainvillea and trendrils of cucurbita. D. Which of the following fish led to the extinction of an ecologically unique assemblage of more than 200 species of cichlid fish in large Victoria of East Africa? Nile perch. C. Here the assertion and reasoning. The embryo with 8 to 16 blastomeres is called a morula. The morula continues to divide and transform into the trophoblast. So C is the right answer. A is true, but reason R is false. B. 14th question. The right answer is B. Both the assertion and reason are true, but R is not the correct explanation of the assertion. Both are true. 15th, Trichoderma species are free-living fungi that are very common in the root ecosystems. They are effective biocontrol agents of several plant pathogens. C is the right answer. The use of chitinase enzyme is necessary for the separation of DNA from yeast cell, but not in the case of Spirogyra. A is the right answer. Okay. Name the functions of each of the following: tassels of corn cob and scutellum. Tassels of corn cob, they are the male flowers that hang at the top of the plant. Their function is to trap pollen grains from the wind to facilitate wind pollination. So, trap pollen grains. They have to trap the pollen grains from the wind to facilitate wind pollination. Scutellum, this is a large shield-shaped cotyledon found in monocot seeds. This is a monocot seed. Its primary function is to absorb nutrients from the endosperm. एंडोस्पर्म से न्यूट्रिएंट को एब्जॉर्ब करना and provide them to the growing embryo. Farmers prefer apomictic seeds over hybrid seeds. Give any two reasons. What are the reasons? Why farmers prefer apomictic seeds over the hybrid seeds? Because they are cost effective is one reason. Then second is trait preservation. Okay, the qualities are ensured, maintained consistently across generation without variation. So therefore, apomictic ensure that the desirable hybrid qualities are maintained consistently across the generation. Define the term amniocentesis. Why has the government imposed a statutory ban in spite of its importance in the medical field? Amniocentesis is a prenatal diagnostic technique where a small amount of amniotic fluid has been taken from the uterus of a woman to detect the genetic disorder or chromosomal abnormalities in the fetus. But it has been banned because the technique was being misused for the female foeticides. People were using it to determine of the unborn child and then illegally aborting the female fetus. Okay, so that is amniocentesis. Name the lymphoid organ that acts as a large reservoir of erythrocytes. Explain its role as a lymphoid organ. The name of the organ is the spleen. Spleen is that organ, and it is a lymphoid organ. The spleen acts as a large filter for the blood. It is a large filter for the blood, and it contains lymphocytes and phagocytes that trap and destroy blood-borne microorganisms, thereby playing a crucial role in the body's immune response. So spleen is that organ. Okay. Suggest how a virus-free healthy plant can be obtained from a diseased sugarcane plant. How can we obtain a virus-free healthy plant from a diseased sugarcane plant? So, suggest how a virus-free healthy plant can be obtained from a diseased sugarcane plant. How can we obtain it from a diseased sugarcane plant? So, a virus-free plant can be obtained through meristem culture. Meristem culture means specifically apical or axillary meristem. Even if a plant is affected with a virus, the meristem remains virus-free because the rate of cell division there is faster than the rate of viral multiplication. So by culturing this meristem in vitro, a healthy plant can be regenerated. So that is in vitro fertilization. Okay. In order to force bacteria to take up the recombinant DNA, they must be made incompetent. Explain how it can be achieved. So it can be achieved through the chemical treatment, or second is the heat shock. Chemical treatment and heat shock. Treating the bacteria cell with a specific concentration of a divalent cation such as Ca2 positive, and heating the DNA is incubated with cells on ice followed by a brief exposure to heat shock and then putting them back on ice. This temporary temperature change forces the bacteria to take up the recombinant DNA. Okay. Differentiate between grazing food chain and detritus food chain. What is grazing food chain and detritus food chain? So grazing food chain starts with green plants. Green plants are the producers. And detritus food chain starts with dead organic matter. Dead organic matter. And what is the energy source? Solar energy is the primary source in grazing food chain, and in the detritus food chain it comes from the dead organic matter, organic waste. Energy comes from the breakdown of organic waste. Okay, so that is the grazing food chain and detritus food chain. The example is the grass, then goat, then man, and here is the dead leaves, earthworm and frog. Okay, ecological pyramids are widely accepted, but they still have some limitations. Write any two limitations. What are the limitations of ecological pyramids? First limitation is the multiple trophic levels. Second is the exclusion of saprophytes. Exclusion of saprophytes. Next question is 22. 22nd question. List any 3 outbreeding devices that flowering plants have developed for cross-pollination and explain how they help to encourage cross-pollination. So what are the devices? The device is dichogamy. This is a maturation of anther stigma at different times. Self-incompatibility, genetic mechanism, and unisexuality. This is the outbreeding devices that flowering plants have developed for cross-pollination. And how it helps? If the pollen is released, if we are talking about the dichogamy, if the pollen is released before the stigma is receptive, or if the stigma becomes receptive before pollen release, self-pollination with the same flower becomes impossible. Okay. And the second is self-incompatibility. It prevents the self-pollen from fertilizing the ovules by inhibiting pollen germination. Then unisexuality. If a plant bears only male or only female flowers, it ensures that cross-pollination is the only way for fertilization to occur. Okay. Then next question is draw a diagram. So you can draw the label diagram. We will go to 24th question. Identify the plant which acts as an immediate ancestor of both ferns and conifers. Which plant is this? Xylophyton. Okay, so this is the plant. Nearest ancestor of flowering plant. Who is the nearest ancestor? Seed ferns. Seed ferns is the nearest ancestor. Name the most primitive group of plants. Primitive group is chlorophyte ancestors. Chlorophyte ancestors. Okay. Xylophyton provides common ancestry to which classes? Ferns and conifers. Common ancestry to ferns and conifers. Name the common ancestor of Xylophyton and seed ferns. Xylophyton and seed ferns का common ancestor है Trachophyte ancestor. Trachophyte ancestor. Name the common ancestor of mosses and trachophytes. Chlorophyte ancestor. So we have already done the objective type of this set 57/5/1, and this paper is available on CBSE board online, and all sets you are seeing, they will also be available. Section A, the answer will be B. Then second question, the answer will be B. Then third, the B. Fourth, D. Then fifth, D. Then sixth, C. Then seventh, statins, D. Eighth, D. Ninth, B. 10th, C. 11th, D. 12th, C. 13th, C. 14th, B. 15th, C. 16th, A. So if you want me to discuss doubt in 16, what is the doubt in 16? The use of chitinase enzyme is necessary for the separation of DNA from the yeast cell, but not in the case of Spirogyra. Fungal cell, set 2 please. Set 2 is not available. Set 2 we have. We have set 2. Okay. Okay, set 2 we will be solving in a short while from now. We will have what is the problem in 16th question, somebody is asking. Here both the assertion and reason are true, and reason is the correct explanation. Fungal cell wall is made up of fungal cyting. 16, 15. We have set 3 also. Yes, we have set 3. We will be talking about set 3 also. Okay. यही होगा 5/3. What is the whole code of the set 3 you are asking? We are solving 57/5/1. 15th का बताओ मैडम. Trichoderma species are free living fungi that are very common in the root ecosystem. They are effective biocontrol agents of several plant pathogens. Here the answer is C. Why C? You are asking. अच्छा, ऑब्लिक 2 का बताना है, तो ऑब्लिक 2 हम अभी कर रहे हैं सॉल्व. पहले ये 15th का पूछ रहे हैं ये लोग, क्यों इसका 15th का सी ऑप्शन है. Reason R is the correct explanation. Assertion is true but reason is false. सी का पूछ रहे हैं. Trichoderma is the fungi. Yes, that are very common in the root ecosystem, and they are effective biocontrol agents of several plant pathogens. So R is false, beta. R is false. Biocontrol agents of several plant pathogens. They are the efficient producers of extracellular enzyme. They are the efficient producers of extracellular enzymes. They are not the biocontrol agents of several plant pathogens. Okay. हाँ, सेकेंड सेट हम अभी सॉल्व कर रहे हैं. सेकेंड सेट आप लोग थोड़ा वेट करें. सेकेंड सेट वी आर सॉल्विंग. Some questions may be different. सेट डी का बताना है. $29 question, 30th question. Which question? 29 and 30th are there in set D only. Set D. Okay. Read the following passage and answer the questions that follow. The process of copying genetic information from template strand of DNA into RNA is called transcription. It is mediated by RNA polymerase. Transcription takes place in the nucleus of cells. In the transcription, only a segment of DNA and only one of the strand is copied into RNA. Why is the strand of DNA with Why is the strand of DNA's polarity transcribed and not the other strand? The RNA polymerase can only catalyze the polymerization of ribonucleotides in one direction, that is 5 to 3. Therefore, it must use the 3 to 5 polarity as a template to ensure the raw new RNA strand is synthesized. Okay, so this is the reason why is RNA required to undergo splicing. Why is hnRNA required to undergo splicing? Mention any two additional processes which hnRNA needs to undergo after splicing to become functional. Heterogeneous nuclear RNA contain both exons and introns. So it is having the introns and exons both. The splicing is required to remove the non-functional introns and join the exons together in a defined order to form functional RNA that can be translated into a protein. Okay? So therefore the splicing is required to remove the non-functional introns and join the exons together to form in a defined order. Okay? So that it can be translated into the protein. Mention the two additional processes which the heterogeneous nuclear RNA needs to undergo after splicing to become functional. What are the additional two processes? Capping and tailing are the two processes which are required. Capping is adding an unusual nucleotide to the 5' and adding adenylate residues to the 3'. Why is the only one strand of DNA transcribed? Give two reasons. One strand is transcribed because of protein complexity. protein complexity, and RNA interference. Okay. Then 38th question. Why are plasmids and bacteriophages used as cloning vectors? Because they have ability to replicate within bacteria cell. Okay, because they have the ability to replicate within bacteria cells independently of the control of chromosomal DNA. Identify the gene in the cloning vector that controls the copy number of the vector. या क्या बोल रहे हैं आप? This is 5/57/5/1 set 1. Identify the gene in the cloning vector that controls the copy number of the vector. This is ORI, that is the origin of replication. It is a sequence from where the replication starts. The restriction site C in the ROB gene, PVU2, it is the standard PBR 322 vector. Yes, the restriction site located within the ROB gene is PVU2. Identify and name the two selectable markers shown in the diagram. What are the selectable markers? This refers to the PBR322 vector. So that is ampicillin resistance gene, AMPR, and the second is tetracycline resistance gene, TETR. These are the two selectable markers, are the antibiotic resistance genes. Name the two restriction sites each in the two genes you have identified as selectable markers. PST and PVU in AMPR ampicillin resistance genes, and tetracycline gene is BAMH and SAL. Okay, so that's all for this set. We will be coming with the next set. Yes, Aditya, you are right. This is the same set you are having. How was your exam, Aditya? Good. And what about your NEET exam? When is your NEET exam? Okay. So that's all about this set. That is the set 2 from the 57/5 series. We'll be having the set 2 in a short while. For the time being, you can check your MCQs. If you have not completed, you can check your MCQs. Or you can check the MCQs in the cbseboardonline.com. That is the site from where we are getting the question papers. Okay. So we are having the second set, cbseboardonline.com. Okay, 57/5/2. Some of the students are asking about the second set. So this is the same question which is there in the first set also. So D is the right answer in this question, both A and B. The paper was moderate, not easy, not difficult. One student was asking about the paper, whether the paper is moderate or the paper is easy. So paper is moderate. Cells of endosperms have 24 chromosomes. What will be the number of chromosomes in the gamete? Two set. Okay, thank you, beta. Answer is 8 here. The number of chromosomes in the gametes are 8. Okay. Any students is having the page number 7 and 8 of this set, please send the page number 7 and 8 of 57/5/2. Please, if you are having, send the page. One student has sent us the paper, but he has not sent the page number 7 and 8. So if you are having, please send, so that your juniors can be benefited from these papers. What we are doing, we are simply uploading the papers for the purpose of your juniors, so that they can see the previous year question papers. So anybody who is having the page number 7 and 8, please send us Okay, thank you. Now moving on to the 7th question. Select the odd option in the context of convergent evolution. So this is D, thorns of bougainvillea and tendrils of cucurbita. D is the right answer here. Monoscus purpurus is a yeast used commercially in the production of which one of the following? Statins. 9th we are having D. This is the same question which is there in the one set. Which of the following ecosystem is most productive in terms of net primary productivity? 10th question. Tropical rainforest, most productive in terms of net primary productivity. Bt toxin genes have been expressed in plants in order to provide resistance against correct answer is C, resistance against third and fourth. Okay, according to IUCN, some of the extinctions include, what are the extinctions which are included? Answer is C again, first, third and fourth. Dodo, thylacine and stellar sea cow. Okay, then the 13th question, both are true. Okay, and R is the correct explanation of A. A is the right answer for 13. Anybody who is having this set, please send the page number 7 and 8 of this set. 6 and 7 page number. Fourteenth question, the answer will be B. And for 15th, the answer is again B. Okay. R is not the correct explanation of A. What about 16? 16th assertion, again B, not the correct explanation of A. Repetitive sequences do not have direct coding function in the genome. Both are true, but R is not the correct explanation of A. Perimetrium, 15th का C. 15th C, they are saying, one of the student is saying 15th C, the endometrium undergoes cyclic changes during the menstrual cycle. Perimetrium contracts strongly during delivery of the baby. Okay, let me check. B is the right answer, beta, not C. Fifteenth. B is the right answer. The myometrium exhibits strong contraction during the delivery of the baby. So, what is reason is not the correct explanation. Both are true, but reason is not the correct explanation. The myometrium exhibits strong contraction during the baby. It is not the correct explanation. Okay. The wall of the uterus consists of 3 layers of tissue: the external thin membranous perimetrium, a middle thick layer of smooth muscle myometrium, and an inner granular layer called endometrium that lines the uterine cavity. So the endometrium undergoes cyclical changes during the method of the cycle, whereas myometrium exhibit the strong contraction. Okay, it is not perimetrium. C is the answer of this. 5 marks please. 5 marks please question. What are 5 marks question? These are the here the label, diagram and structures you are having. That will. Okay, let us see. Let us see. Work out the separate monohybrid. No, we are not having 3 right now, beta. We are working on set 2. We are working on set 2. This is set 2. Five marks question. Yes. Set 1 का 5 marks question. Okay. Set 1 के 5 marks question में क्या है बेटा? देखते हैं, रुको. Explain the events of fertilization and implantation in an adult human female. Set 1 5 marks question. So events are ovulation first, then sperm transport and capacitation. I will fail, ma'am. I knew it. Only 6 MCQs were right, but theory was very, what are you saying? Theory was right, was very, I didn't know that I will get number or pass. Don't panic. Yes, Reena, what you are saying? You are saying, na, set 1 and 5 marks question. 32 question. Okay. Work out separate monohybrid crosses up to F2 generation between 2 pea plants and 2 anti-herinum plants. This is set 1, 32A, both having contrasting traits with respect to the color of the flower. Comment on the patterns of inheritance in the crosses carried out in such 2 cases. Okay. Set 2 MCQ we have already done. Set 2 MCQ we have done, beta. We have done the set 2. 32 questions he is asking. Reena is asking about the 32 question. So the complete dominance of the monohybrid cross. The parents are having violet flowered, that is VV, and white flowered, that is small v, small v. F1 generation, all VV. F2 generation, VV into VV. Genotype, phenotype, 3 violet, 1 white. One white, VV. Inheritance pattern, complete dominance, Mendelian. And then second is the monohybrid cross, antirrhinum, flower color red white. And here the inheritance pattern is incomplete dominance, non-Mendelian pattern where heterozygotes show intermediate phenotype. Yes, incomplete dominance is here. Incomplete dominance is here in the second case where non-Mendelian is there and where the heterozygotes show intermediate phenotype. Okay, is it clear? Reena, you were asking about the 32nd question. What about the set 2? You are saying the set 2. We have done the set 2. We have done set 2. Any other doubt? First is the complete dominance and second is the incomplete dominance in 32A. How does a chromosomal disorder? You are a private student. I failed compartment previous year in 2025. So in 26 as a private student, if I fail this time, then I have to do 12th second time. Please tell me. What to do in this case? As a private student, if you are failing second time, then you have to repeat 12. That is your question. How will you are a private student, no? Yes, you have to repeat the class 12th if you fail or receive a compartment. If failed in one or two subject, you can take immediate compartment exam to pass the same year. You can take the immediate compartment exam to pass in the same year. If failed in more than two subjects, you must re-enroll as a private candidate for all subjects the following year. Yes, you have to repeat all the subjects. If you fail in one subject, you can apply for the compartment, which is usually going to be held in July or August. Okay. And if you are failing in two or more papers, you must reapply as a private student for the next academic year. But if you are passed in practical but failed in theory, so you can carry forward your practical marks as explained in the CBSE website. You can go and check the CBSE website. They have the clear points over this, what you have to do. Okay, any other doubt you are having, or just we can finish this? We have solved the two sets, that is 57/5/2 and 57/5/1. Both sets are available on the cbseboardonline.com. If you want, you can check that. Okay. So that's all. If you have any doubt, you can comment.